Hello everyone, welcome back to one more video session. In this session, I'll be speaking about another very interesting as well as very important for your board as well as NEET examination. The experiment name is Biochemical Characterization of Transforming Principle. It is the continuation of Griffith's transforming principle conducted by three famous scientists, the Avery, MacLeod and McCarty. Just to know which is exactly the transforming principle which caused the transformation of R strains into S strain, which caused the death of the mice. Mice got pneumonia and finally died. Correct, we know about it. That's what we have studied in Griffith's transforming principle. Now we have to find what is the transforming principle which caused the transformation of R strains into S strain. Okay, so here you could see I have written biochemical characterization. First of all, what is a biochemical? You take a cell and you see many biochemicals like lipids, sugars, polysaccharides, carbohydrates, and also proteins, DNA and RNA. Now out of these 6 different types of biochemicals, which is the transforming principle or which is the genetic material? Okay, so they conducted the experiment by taking heat killed S strain, and you could see here, yes, before the experiment the assumptions were it could be protein, DNA or RNA is the transforming principle, or maybe it could be the genetic material. Out of 3, any one could be the genetic material. After the completion of the experiment of this biochemical characterization, the 3 scientists came to the conclusion that DNA is the transforming principle. Let us see how. Very simple. You could see here, they took the heat killed S strains. Obviously, they killed it by heating. Okay, and after that, they just put it inside a test tube containing some solution. And after that they went for centrifugation, that rotation method. As a result, they could separate lipids as well as sugars. That's what I have written here. They removed the lipids and sugars because they knew that lipid and sugar could not be a genetic material or transforming principle. That's it, they removed it. What is remaining now? Out of 5, now we have 3 biochemicals, that is proteins, DNA or RNA, and RNA, not RNA. Okay, or RNA, it is RNA. Okay, out of 3. Now they took 3 different test tubes and just put this solution into each test tube like that. You could see here all these containing the heat killed S strains, that's it. Now what they did, they just put a particular type of degrading or digesting enzyme. There are so many enzymes which can destroy the RNA, DNA or protein, but these enzymes are particular in or specific in function. For example, see here, like for this particular test tube they added proteinase enzyme. The mechanism or the function of the proteinase enzyme is to kill or not kill, destroy the entire protein, whatever the protein which is present within the S cells, heat killed S cells. Obviously they will remove the proteins. Okay, in this test tube they added the RNase, that means it will destroy or digest the RNA. And here they added DNase, which can destroy the DNA or digest the DNA. Like that, okay, they put a specific type of enzyme into each test tube. You could see here the proteinase number 1, RNase number 2, DNase number 3, like that. That is the second step. And after that, they took a Petri plate or Petri dish. In that, they grew the live R strains, like that. You have to imagine Petri plate, 3 Petri plates. In each Petri plate, what is there? The R strains are there, the live R strains are there. Then they just pour this entire solution into the Petri plate. They poured it. Okay, so that's it. Now, because as there is no protein here, I have written no protein because the protein has already digested the proteins. What is remaining now? What are the biochemicals which is remaining in this test tube? DNA as well as RNA. Okay, and after that the mixture of heat killed S strain plus R strains, and obviously they expected transformation. That thing happened. Okay, that means we can come to the conclusion that the protein is not the transforming principle. It could be RNA or it could be DNA. That is actually the thing happened. Whatever the R strains were there got transformed into S strain. That's it. Because the DNA and RNA still it was there, not the protein. So we can come to the conclusion that the protein is not the genetic material or not the transforming principle. That's it. So no, here you could see transformation has occurred. Okay, live S cells appear. They could remove the S cells. live S cells. And in the second case, they added RNase, and obviously no RNA is removed now. So what is remaining now? Protein as well as the DNA. Obviously they came to the conclusion that protein is not the transforming principle. What is remaining now? DNA. Okay. And again they got the same result, that is R cells got transformed into live S strains. So that is the thing. How did they know that okay it is transformed? Why? Very simple, by looking into the colony where they developed smooth colonies, shiny smooth colonies. That means they have developed polysaccharide coat around their cell wall. That's the thing happened, correct? So in these two cases, transformation has occurred. The R strain got transformed into S strain. That is the thing occurred. In the third step, when they added the DNase into the solution containing heat-killed S strain, that means the DNA is now removed by the DNase activity, that is digested. What is remaining? Protein as well as RNA. And when they mixed this heat-killed S strain solution into the live R strains, which is in the Petri plate, they did not get the kind of polysaccharide coat, or they did not see the smooth shiny colony developed within this Petri plate. That means transformation has not taken place. That is the thing happened here. Okay, so they did not see the transformation of live R strains into S strain because there is no DNA. So they came to the conclusion that okay, so DNA is the transforming principle, what the Griffith said. The Griffith did not know about that DNA actually that caused the transformation of R strains into S strains. But because of this experiment, these scientists they came to the conclusion that yes, it is the DNA which caused the transformation of the R strain. So this is actually all about biochemical characterization of principle, and they completed their experiment by 1944. But still the people or the scientists they did not even believe that okay, so DNA is the genetic material. They were not actually convinced that DNA exactly the genetic material. So they had to wait for another 8 more years until Hershey and Chase conducted another experiment that was the proof that gave the proof that the DNA is itself the genetic material. Okay, so this is the second experiment. Please remember it. It could be asked for 5 marks and also obviously for the NEET examination. Okay, so hope you understood this concept.